In 2016, a Japanese scientist named Yoshinori Osumi won the Nobel Prize in Medicine for discovering something that had been happening inside every cell of your body since the day you were born. A process so fundamental to human survival that without it, your cells would choke on their own waste within days. The discovery was autophagy, a word that comes from the Greek for self-eating, and what it describes is your body's built-in cellular recycling system, a sophisticated internal cleanup crew that identifies damaged proteins, broken down organelles, toxic debris, and even invading bacteria inside your cells, wraps them in a membrane, and delivers them to a structure called a lysosome where they're dissolved, broken into raw parts, and either discarded or repurposed as building material for new, healthy cellular components. Think of autophagy as the difference between a house that gets cleaned every week and a house where the garbage never gets taken out. In the clean house, the surfaces stay clear, the air stays fresh, and everything works the way it should. In the other house, the trash piles up, the mold spreads, the systems start breaking down, and eventually the structure itself becomes unsafe. That second house is what happens inside your cells when autophagy slows down. And here's the part that matters most for anyone watching this. Autophagy naturally declines with age. By the time you're past 60, your cellular cleanup crew is working at a fraction of the speed it had in your 20s, which means damaged proteins accumulate faster, toxic debris builds up more aggressively, and the risk of chronic disease, including cancer, Alzheimer's, cardiovascular disease, and diabetes, increases precisely because the cells can no longer keep themselves clean. But here's what Osumi's Nobel Prize-winning research and the explosion of autophagy science that followed it have revealed. And this is the part that changes everything for anyone over 60. Certain foods contain specific, well-characterized compounds that can reactivate autophagy, essentially waking up the cleanup crew and putting it back to work even when aging has slowed it down to a crawl. Not through some vague detox mechanism that you'd find on a wellness blog next to advertisements for juice cleanses, but through precise molecular switches that researchers have mapped in extraordinary detail. Switches with names like AMPK, MTOR, SIRT1, and PI3K that control whether your cells are in growth mode or cleanup mode at any given moment. When you understand that these switches exist and that specific foods can flip them, you stop thinking about nutrition as calories and vitamins and start thinking about it as cellular programming, because that's exactly what it is. So today I want to show you 4 foods that flip those switches, each one working through a different pathway, each one backed by published research from major universities, and each one available at any grocery store for a few dollars. Food number 1 is green tea, and it activates autophagy through the same molecular switch that fasting does, except you don't have to stop eating. Green tea contains a polyphenol called EGCG, epigallocatechin galate, that is one of the most extensively studied autophagy activators in the entire nutritional science literature. What EGCG does at the cellular level is remarkable. It activates an enzyme called AMPK, which acts as your cell's energy sensor and master regulator of metabolic cleanup. When AMPK switches on, it simultaneously inhibits a pathway called MTOR, which is the signal that tells your cells we have plenty of resources, no need to recycle anything. And when MTOR goes quiet, autophagy ramps up. It's the same pathway that gets activated during fasting, which is why fasting has been so extensively linked to cellular renewal, except EGCG can trigger this cascade while you're still eating normally, giving you a way to stimulate the cleanup without the hunger and the difficulty of prolonged food restriction. A study published by researchers at Rutgers University found that EGCG stimulated autophagy in liver cells specifically, increasing the formation of autophagosomes, the membrane-wrapped garbage bags that collect cellular debris, and promoting the clearance of fat droplets that had accumulated inside those cells. Another study published in the journal Oxidative Medicine and Cellular Longevity showed that prolonged EGCG supplementation reduced markers of DNA damage, suppressed cellular senescence, the state where damaged cells stop dividing but refuse to die, clogging up tissues and releasing inflammatory signals, and promoted autophagy-related gene expression in aging cells. This suggests that green tea doesn't just trigger the cleanup, it directs it toward the specific types of cellular waste that are most dangerous in the context of metabolic disease, cancer development, and age-related decline. 3 to 4 cups of high-quality green tea daily, steeped in water, just below boiling for 3 to 5 minutes to maximize EGCG extraction without destroying it, is the range most commonly associated with meaningful autophagy activation in the research. And because EGCG is simultaneously a powerful antioxidant, anti-inflammatory, and neuroprotective compound, the benefits extend well beyond cellular cleanup into virtually every system in the body. Food number 2 is one that most Americans walk right past in the produce section, and it contains a compound that may be the single most potent dietary autophagy activator ever identified. Mushrooms, particularly varieties like shiitake, maitake, and even common white button mushrooms, are among the richest dietary sources of a compound called spermidine, a naturally occurring polyamine that has generated enormous excitement in the longevity research community over the past decade. Spermidine activates autophagy through a mechanism that's distinct from EGCG, rather than working through the AMPK-mTOR energy sensing pathway. Spermidine directly influences a process called acetylation, which controls whether autophagy genes are switched on or off at the level of your DNA. It essentially unlocks the genetic instructions for cellular cleanup and makes them more accessible to the cell's machinery, allowing autophagy to proceed even when other signals might be telling the cell to keep its recycling systems idle. The research on spermidine is some of the most exciting work happening in the longevity science right now. A landmark study published in the American Journal of Clinical Nutrition followed a large population of over 800 adults for 2 decades and found that those with the highest dietary spermidine intake had significantly lower rates of cardiovascular disease, cancer, and all-cause mortality compared to those with the lowest intake. And the magnitude of the protective effect was comparable to what you'd expect from a regular exercise program, which puts a simple dietary compound in the same league as one of the most powerful health interventions known to medicine. Animal studies have been even more dramatic. Spermidine supplementation has been shown to extend lifespan by up to 25% in certain models, improve cardiac function in aging hearts, reduce neuroinflammation in aging brains, and preserve memory and cognitive function. And in every case, the proposed mechanism traces back to enhanced autophagy, clearing out the damaged cellular components that would otherwise accumulate and drive disease. Beyond mushrooms, spermidine is also found in meaningful quantities in aged cheeses like cheddar, Gouda, and Parmesan, in green peas, in wheat germ, in broccoli, and in fermented soybean products like miso and tempeh. But mushrooms remain one of the most concentrated, most versatile, and most accessible sources for the average American grocery shopper. Incorporating a generous serving of mushrooms several times a week, sautéed with garlic and olive oil, roasted until the edge is crisp, added to soups, or mixed into omelets and stir-fries, is a simple, inexpensive way to boost your spermidine intake and give the autophagy process the molecular push it increasingly needs as you age. Food number 3 is turmeric, and its active compound doesn't just reduce inflammation, it directly commands your cells to start recycling their damaged parts. Curcumin, the bright yellow compound that gives turmeric its distinctive color, has been studied for decades for its anti-inflammatory and antioxidant properties, but what's become increasingly clear from research published in the last few years is that curcumin is also a powerful autophagy inducer, working through yet another molecular pathway, TPCM, the PI3K/AKT/mTOR signaling cascade. In simple terms, curcumin suppresses the signals that tell your cells to keep growing and consuming resources without cleaning up after themselves, and it amplifies the signals that trigger the internal recycling program. The result is that damaged proteins, dysfunctional mitochondria, and accumulated cellular waste get identified, wrapped up, and sent to the lysosome for breakdown. Exactly the same cleanup process that Osumi won his Nobel Prize for elucidating. What makes curcumin particularly interesting in the context of cancer prevention is that autophagy serves as one of the body's most critical quality control mechanisms, a system that can identify and eliminate precancerous cells before they have a chance to multiply into a tumor. Every day, your body produces cells with DNA errors, cells with damaged mitochondria, cells that have started behaving abnormally. And under normal circumstances, autophagy flags these cells, breaks them down, and recycles their components before they can become a threat. When autophagy slows down with age, that surveillance system weakens, and damaged cells that would have been caught and destroyed in your 30s start slipping through the net in your 60s and 70s. Curcumin, by enhancing autophagy through the PY3K/AKT/MTOR pathway, essentially reinforces the net, helping the body catch and destroy precancerous cells at a stage when they're still manageable, which represents a profoundly different approach to cancer prevention than anything you'd find in a pharmacy. The practical challenge with curcumin is absorption. Your body doesn't absorb it efficiently on its own, which is why most of the research uses enhanced formulations. The simplest kitchen solution is to always consume turmeric alongside black pepper, which contains a compound called piperine that increases curcumin absorption by up to 2,000% and with a source of fat, since curcumin is fat soluble. A teaspoon of turmeric stirred into a warm cup of milk or broth with a pinch of black pepper and a drizzle of olive oil is one of the most bioavailable ways to deliver curcumin to your cells on a daily basis. Food number 4 is dark-colored grapes and berries, and the compound that gives them their deep pigment is also the one that tells your aging cells to start cleaning house. Resveratrol, the polyphenol found in the skins of red and purple grapes, in blueberries and cranberries, and in smaller amounts in peanuts and dark chocolate, has been making headlines in longevity research for over 2 decades, initially for its cardiovascular benefits, but increasingly for its role as an autophagy activator. Resveratrol works by activating a protein called SIRT1, which is sometimes called the longevity gene because of its central role in regulating cellular stress responses, DNA repair, and metabolic efficiency. When SIRT1 is activated, it triggers a cascade that includes AMPK activation and MTOR suppression, the same fundamental pathway that fasting activates and that EGCG from green tea also stimulates, but through a slightly different entry point, which means the effects can be additive when these foods are consumed together. A landmark study from Harvard Medical School found that resveratrol activation of SIRT1 mimicked many of the molecular signatures of caloric restriction, the only intervention consistently shown to extend lifespan across multiple species in laboratory settings without requiring any actual reduction in food intake. The cells of resveratrol-treated animals behaved as though the body was in a fasting state even when it was eating normally. Autophagy ramped up, mitochondrial function improved, inflammatory markers dropped, and the accumulation of damaged proteins that contribute to aging and neurodegeneration slowed significantly. Subsequent research from the National Institute on Aging has confirmed that resveratrol activates many of the same protective pathways as caloric restriction, and while the effect in humans is still being quantified through ongoing trials, the mechanistic evidence from cell studies and animal models is robust enough that the longevity research community considers it one of the most promising dietary compounds currently under investigation. The richest food sources of resveratrol are dark-skinned grapes eaten with the skin on, since resveratrol is concentrated in the skin, not the flesh, fresh blueberries, cranberries, mulberries, and to a lesser extent peanuts and dark chocolate. A daily handful of dark red or purple grapes or a cup of mixed berries combined with the green tea, mushrooms, and turmeric we've already discussed creates a multipathway approach to autophagy activation that no single food could ever achieve on its own, each one entering through a different molecular door, and together ensuring that the cellular recycling program your aging body needs most gets the broadest, most comprehensive reactivation possible. Here's what I want you to understand after everything we've covered today. Autophagy isn't a wellness trend, it isn't a marketing buzzword, and it isn't something you need an expensive supplement to unlock. It's a Nobel Prize winning biological discovery that explains at the deepest level of science currently understands how your cells stay healthy, how they defend against cancer and neurodegeneration, how they slow the aging process, and how they clear out the molecular garbage that accumulates with every passing year and every metabolic process your body runs. Green tea for AMPK activation and MTOR suppression, mushrooms for spermidine-driven gene unlocking, turmeric for PI3K/MTOR, pathway modulation and cancer cell surveillance, and dark grapes and berries for SIRT1-mediated longevity signaling. These 4 foods, eaten regularly and consistently as part of your normal meals, provide your cells with 4 distinct molecular keys to restart a process that aging has been gradually and silently shutting down since your 30s. And here's what makes this approach so powerful. Because each food activates autophagy through a different molecular doorway. Combining them doesn't just add up, it compounds. You're not pushing one button 4 times. You're pushing 4 different buttons that each activate a different arm of the same cleanup system, giving your cells the broadest possible signal to start recycling, repairing, and renewing themselves. The cleanup crew hasn't quit. It's just been waiting for the right signal to get back to work, and now you know exactly which signals to send. Share this with someone who needs it. Subscribe so you don't miss what's coming next. And as always, take care of yourselves. I'll see you in the next one.